Thank you very much for the chance to speak to you today. I'm here to talk about the only group of viruses Eugene didn't talk about this morning. Which are? Yes. Well, having Eugene Koonin as your pitch man for your talk is a little intimidating. Okay, so these are the circular, rep-encoding, single-stranded DNA viruses that infect eukaryotes. That may sound like I'm narrowing down the universe more and more, but this is an arbitrary distinction. There's a reason to study this group as a cohesive group, and that's that they have a homologous ORF that is shared across pretty divergent genomes and a wide range of different eukaryotic hosts. So that's why we consider them a group. We know a lot more about this group than we did 10 years ago because the 529 polymerase that's already been mentioned a few times in the symposium preferentially amplifies single-stranded DNA templates, and rolling circle amplification works best on circles. So if you have something that's a CREST virus-like genome in your sample, you will see a big representation of it in your eventual sequencing results. And while viral metagenomic techniques have completely transformed many groups that are well represented in this room, CREST viruses have been no exception. This has been a decade of discovery and a reimagining of the importance of this group. In the 9th version of the ICTV in 2011, there were only 3 recognized families of CREST viruses. They infected hosts that were economically important for food security, typically, the Circoviridae, Geminiviridae, and Nanoviridae. Now, as of the most recently accepted proposals, there are 6 established families, many more genera within the established families, and we know that the host range of CREST viruses is much wider than previously thought. We're infecting heteroconts, we're infecting fungi, and there's a lot of big black plus signs up here on the slide because most of these new sequences coming into GenBank don't have a definitive host. They were isolated in association with some host. So we don't know if the CREST virus is infecting that particular host, infecting something that host ate, infecting something else in that host's holobiont. So we know that our diversity of hosts that are infected by CREST DNA viruses is likely much larger than even what we know so far, but we can't be definitive about it because we don't know that much about these viruses' ecology. I'm going to talk today about 2 different projects my lab's been working on to try to understand the evolution of the CREST DNA viruses better. The first is trying to create a robust genealogy of that one homologous ORF, that replication-associated protein. Rep is not a polymerase. It just assists in rolling circle replication. And that project is being led by a senior graduate student in my lab, Lila Zhao. And then we've also been fooling around with different ways to try to assist in definitive classification of novel virus sequences. And I'm going to talk about the work of a postdoc in my lab, Eric Lavington, and a first-year graduate student in my lab, Alvin Crespo. When we set out to create a genealogy for the rep protein, one of the things that my student Lila asked is, what kind of amino acid substitution matrices are others using when they're looking at the diversity of rep across CREST DNA viruses? Over the course of 10 years, and all of the rep amino acid trees that have been published, these are the different kinds of substitution matrices or methods used to create those trees. Overwhelmingly, researchers just use whatever ProTest tells them to use, and that ends up being LG, the most modern empirically derived general out-of-the-box substitution matrix you can get. That's wonderful. Sometimes different matrices show up as being important. For example, RTRev here, which is a bespoke matrix developed for retrotranscribing elements, not infrequently shows up as being one that you would use to describe circular single-stranded DNA virus evolution. I wish we could say that was the inspiration for it, but we had the idea anyway. If what we really care about is making the best rep tree possible, let's see if a bespoke matrix for CREST DNA viruses can get us there better than some of the general matrices that have been developed. And so Lila did this by assembling a giant data set of rep sequences from all of the NCBI ref-seqs for the CREST DNA viruses. It's about 900 sequences. We have a few extra sequences thrown in from novel genomes that are from our collaborators, the Breitbart Lab at the University of South Florida. As they're getting, the number used to be larger, they're publishing things, it's getting into NCBI. We're not hiding too much data from you guys. After assembling all of the ref-seqs, these are amino acid sequences, she aligned them with muscle. We had a lot of insertions and deletions because this is a divergent protein that's been evolving for a huge amount of time. She used TrimAL to determine a set of sites that were well represented across the sequences. We ended up with a final alignment that's around the average length of a rep sequence of about 370 amino acids in our alignment. We ran ProTest to see what it thought the best matrix to describe the evolution of these proteins would be, and they said VT, which was a matrix we hadn't heard of until 2016, when a bunch of papers from Arvind Varsani's lab started being best described with the VT matrix. It's another empirically derived general matrix. Then we split the data set up into 50-50 halves. We jackknifed it into a training data set, which we used to develop potential CREST DNA matrices. We used two different programs for this, FastMG and HiFi, to optimize a substitution matrix based on just half of our rep data set. The other half of our data set was our test data set. We built a tree with that data set using the optimal algorithm from ProTest. And then we put our test tree and our training data set matrices into PAML and asked PAML to look at the fit and the likelihood fit of our data set and matrices to the various trees that were the test trees. And we did that 10 times. So we have 10 different independently derived training data sets with which to find our own CREST DNA matrix. And we didn't just do matrix fitting in one way. Starting with the assumption of either the VT matrix, which had been the optimal for our whole data set, or the LG matrix, which was a pretty good general matrix used widely across CREST DNA tree building. We optimized in two different programs. Each training data set put 4 possible CREST DNA optimized matrices into our alignment, and we also checked the fit of VT, LG, and that other bespoke matrix RT-Rev down there at the bottom. Our results are shown here. Darker colors means better fit with likelihood, and FastMG, working off of the VT matrix, was pretty much the best call across the various groups. We now have 10 best matrices for the individual training data sets, and we have to pick one of them to be our potential CREST matrix. We were reassured that as long as we were picking amongst the best ones, it was going to be about the same. This is a heat map of Pearson correlation coefficients from the different substitution matrices. Up here, we have our general matrices, and then the FMG fit matrices and the HiFi fit matrices. The FastMG matrices are all pretty similar. We could have picked any of them and gotten probably about the same matrix. As it happened, we did a test of each of the 10 best against all of the different test data sets. FMG VT number 10 was the best fit. So that becomes what we're calling the CREST matrix. And if you're not used to looking at amino acid substitution matrices, this bubble chart telling you the likelihood of interchange of different amino acids probably doesn't mean that much to you. Perhaps it means more to you if we compare it to some of the other matrices you may use in your own phylogenetic tree building. A blue color indicates that the CREST matrix has a lower likelihood of interchange of the amino acids. A brown indicates a higher likelihood of interchange than the other matrices. And when we compare CREST to RT-Rev, hey, it's a lot slower than those retrotranscribing viruses, but on average, it's faster than some of our general substitution matrices that were derived on very large data sets, but typically of exclusively cellular protein sequences. We think we have strong evidence now that our CREST matrix best describes the evolutionary patterns in our rep alignment, but you don't have to just look at our CREST trees. We're going to show you trees also created with LG and VT matrices as well. What I'm going to show you here are rapid bootstrap Raxamel trees. These are trees that have over 900 taxa. They're completely illegible. The colors are just showing you the families that the virus sequences are associated with. And bright pink, which will be important, are unclassified to a family at this time. There's lots of sequences in NCBI that are not yet properly assigned to a family or genus in ICTV, as Rodney just showed us. And purple is a group of viral elements that group with the CREST DNA viruses that I haven't told you about yet, which are alpha satellites that are sometimes found in association with plant-infecting CREST DNA viruses. Let's look at just the crest tree as a cladogram, so we can have a bit of a better view at this, but I'm going to talk about the results of all 3 trees at first. Same coloring scheme for the families of viruses as in the previous slide, but now you can see some of the more deep branching patterns. Unfortunately, we don't have a lot of support for these deep branching patterns. Not surprisingly, it's a very divergent protein. Here are little tiny black dots showing you all of the nodes supported by greater than 50% bootstrap values, mostly towards the tips, a couple of them venturing deeper in. But yes, it's very difficult to have very robust support for very deep branching in any protein phylogeny going down this far. Something that all of our trees agree on. When the Nanoviridae, this group of segmented single-stranded DNA viruses of plants, sits within alpha satellites, which also are found in plants. If you make trees with smaller numbers of sequences from characterized families, you're just using a few representative sequences, most of them show that these are sister groups. We're not the first people to say, hey, this viral family sits within a viral element clade, but we got relatively nice support for that, pretty weak bootstrap values, but again, this is going pretty far back in evolutionary time. Much stronger support for something that makes total sense, but we haven't seen a published tree support it yet, which is that the family of Genomic, this newly classified, very diverse group, we only have one cultured member of it that infects fungi. It is a sister taxon of part of the Geminiviridae family, the group Mastrovirus. This makes total sense because mastroviruses and all of the genomoviruses have a spliced rep. They actually cut out an intron in order to make the rep, the final rep. That means that potentially this was a single ancestral intron insertion that is shared by a group of Geminiviruses and all of the Genomic that have been identified to date. And that's great bootstrap support for that particular conclusion, and that bootstrap support is there across all of our 3 trees that we're comparing. All of the trees we're looking at show that the taxonomy within the Cercoviridae is going to get more complicated soon. Cercoviridae just underwent a taxonomic revision in 2017, but if you look in the tree, it's the mustard color, yellow colored one here, there's lots of unclassified viruses popping up in close relationship to the cercoviruses. There are only 2 known genera within the Cercoviridae at this point. They're down here, they're well separated. We don't see this novelty falling neatly within the groups of the 2 genera within Cercoviridae. One nice new thing that we've seen by using the CREST DNA matrix is that we're seeing a relationship between 2 very recently codified families, the Smacoviridae and the Bacillodiniviridae. Am I saying that correctly? Does anyone know how to say it? It only got approved last month. I'm not that behind the times. But Bacillodiniviridae is over here in red. Smacoviruses are over here in navy. In all 3 of our trees, there was always one Smacovirus that would group tightly with the Bacillodiniviridae, but the crest is the only one that's bringing the entire groups closer together. So this is an ongoing project in my lab. If anyone has any complaints or suggestions of things we should be doing differently, this would be a great time at the end of the talk to suggest it because these trees are hot off the presses. Because we're comparing our results to the wider literature, most of the trees that have been published trying to look at the total diversity of rep within the CREST DNA virus world select down to just a few representative sequences. They focus on the fact that there's a lot of diversity here that we don't know what to do with yet, but that misses out on the fact that there's intermingling between our established group viruses and these unknown viruses, or even viruses from different families. That intermingling could indicate misclassification, or it may not indicate that at all, because horizontal gene transfer is very possible within the ssDNA viruses. Recombination is a large source of variation for these viruses, and based on the classification parameters, or that are often whole genome nucleotide percent identity, you can have a rep protein that looks more like family A and still be a member of family B. It's just lovely evidence of horizontal gene transfer. So we're hoping to suss out which of our intermingling sequences are potentially misclassified and which show strong support for a wide-ranging horizontal gene transfer. I want to switch gears now from talking about the rep genealogy to talking about the fact that the diversity of viruses that we're trying to organize here, most of them are known by their sequence only. That means that typically the classification to a family or genus is done by percent nucleotide identity, often on the whole genomic sequence. And very important for classification of cross-DNA viruses is the genomic orientation of the ORFs. So I'm showing you a begomavirus. It's a plant-infecting cross-DNA virus. It has the coat protein here encoded in the virion sense. It looks like the messenger RNA in its genomic form. And the rep is encoded in antisense. That means it's ready to be transcribed into messenger RNA as soon as it gets into a cell. That's an important factor for determining what genus within a family, for example, certain cross-DNA viruses should be in. The problem is that we can get whole genomic classification done pretty well just by blast searches and the like, but determining genomic orientation is a bit trickier because a lot of the time it's determined by this origin of replication up here, the stem loop that's very important in how rolling circle replication works. There is definitely a stem, and there's a loop that has a 9-base motif. The loop is bigger than 9 bases. And we used to think of these motifs as being really invariant. You know, all the begomaviruses have exactly this 9-nucleotide sequence. All the circoviruses have exactly this one or that one of these 9-nucleotide sequence. And as the diversity of these viruses has grown, I think you can see we've gotten a much broader toleration of sequences can be recognized as the origin of replication. So much broader, so broad, in fact, that the complement of some of these non-nucleotide sequences, especially when you take into account some of the other bases in the origin of replication, you're not flipping just these 9, you're flipping 8 of them plus 1 from the loop that I haven't listed up here, could pass for an origin of replication. And that means that we're losing our ability to be sure of what orientation a novel genome is in. We're not characterizing in the lab, so we can't follow up and do some experiments to figure it out. We want to know if there are things we can do outside of the typical nucleotide sequence homology and, hey, does this origin of replication look right to you, that can help us classify these viruses. Our first approach that was separate from nucleotide similarity, we actually undertook the project to try to find novel motifs in the rep protein sequences that hadn't been noticed before by humans. We took a machine learning approach to do this. It ended up working terribly, but told us something cool that's nucleotide sequence free. And then I'm going to talk about a project that's finally gotten some traction in my lab after a lot of false starts of looking at codon bias to help us determine strand orientation in cross-DNA viruses. So machine learning classification, we took the same data set as Lila had for her rep tree building, except we only took viruses that had been classified by ITCV to a official family or genus, but we worked from that starting data set. And we tried 3 different machine learning techniques to sort the viruses to their appropriate genera. We tried a multinomial naive Bayes approach, which allows you to just read in. All of these methods allow you to look at strings of letters without keeping positional information, unless I tell you otherwise. And 2 different kinds of support vector machine classifiers, one with the typical linear function to try to determine what genera should be where, and one with the more complicated radial basis function. We tried putting in data sets that were nucleotides, unaligned nucleotides, nucleotide engrams, that's single nucleotides up to 12 mers in sequence. Codon data sets, primary amino acid sequences, unaligned, amino acid engrams up to 12 mers, and aligned rep sequences from the Luz data set. I'm showing you on the y-axis here the F1 score is a measure of the accuracy of the model in correctly calling all of the sequences to genus. And these are the results of 10,000 jackknife simulations where we took half the data set to train the classifier and tested it on the other half. If you're using nucleotides, nothing's really that great as soon as you get up to the level of codon. When you get to the connection to the amino acid, pretty much all of the methods work pretty well. You don't have to be aligned in order to get good classification to genus. And excitingly, unaligned amino acids, just the strict primary sequence of the rep protein, worked pretty well. In fact, in our final trained linear support vector classifier, it's better than 96% right at doing this against the full data set. That's nice, because even if it's just telling us what other people know from sequence similarity, it came at it from a completely different perspective. What it's using to do this is the amino acid content of the rep. It doesn't care what order those amino acids are in. It's just saying, oh, you're 5% isoleucine, that helps us figure out what you should be. So this is a complementary method, never to replace actual looking at sequence homology, that tells us from another perspective that we're getting the right kinds of classifications. But that was a good consolation prize from a project that failed to give us any n-grams that were of use for classification. In terms of validating this support vector classifier approach, we started the project before the taxonomic revision of the Cercoviridae, and had to update our model after a number of different viruses changed places, and that explained a lot of what was going wrong with our classifier before. So here in March 2017, again, we have accuracy on the y-axis here, we were really good at predicting what circoviruses were. We were pretty bad at predicting what cycloviruses were. Turns out a lot of labeled cycloviruses actually were circoviruses. They fixed their classification. That fixed our problems. So nice verification that we're finding the right kinds of things. We just didn't know that's what was going wrong here originally. I'm more passionate about this next bit of data, and that's why I'm presenting it, even though we haven't gotten to the solution for very long. This has been something that was developed this semester by one of our first-year students. We have, in single-stranded DNA viruses, a known cytosine to thymine substitution bias, and it ends up causing a strand-specific codon bias. I'm not going into all of the data that support that conclusion. I'm presenting data from a published paper, so hopefully you will trust me on that. So what that means is, for example, in this Bogoma virus, we have the coprotein in sense, the rep in antisense, and the fact that cytosines turn into thymines more often than all of the other base substitutions, not combined, but it's just the most frequent one of the 12 base substitutions, means that we see an accumulation in third positions of thymines in the coprotein and A's in the rep codons. And that's because those A's correspond to T's in the anticodons that are actually on the virion sense in the encapsulated genomes. We have been looking for a way to use this C to T strand-specific bias to help us figure out what orientation the genomes are in for some time. But we've been, no, I'm sorry, I'm going to give you some data. Believe me on this. We looked at 15 really well-sequenced Bogoma virus species a few years ago, about 5 years ago. In GenBank, they all infect dicots. If you look at the most frequently used codons for the coprotein, they're not necessarily exclusively in NNT, but we got a large representation down here in NNT. If you look at the rep, we have some NNT preferred codons. It happens to be the dicots themselves prefer to use NNT codons a lot of the time, but we have that enrichment of NNA codons there corresponding to the TNN in the virion sense. This is something that is a signal, not just in Bogoma viruses, but we've been seeing that in other viral groups of CREST DNA viruses in my lab. But we have failed to get anywhere using existing codon bias metrics. We tore codon deviation coefficient apart, all 500 columns of data that you get from it, and we were unable to do something that robustly looks at an ORF and can tell you, based on its codon bias, I think this is a virion sense, or I think this is a complement to virion sense. However, my student Alvin said, hey, a lot of the time you make me look at Ambasense genomes, how about a hypergeometric distribution? I can count all of the codons that are in this genome and then break it down, and typically in circoviruses, you're dealing with two major ORFs, rep and CP. What's the probability I get all the T-ending codons I want to see before I reach the end of my protein with one ORF versus the other ORF? That's working really, really well. It also was successful at finding all of the misclassified cycloviruses. It found all but it found 23 of them, it missed one of them. So it's doing pretty well at finding the correct orientation of the genome just on that alone. Interestingly, it's implicating that 3 circoviruses might be in the opposite orientation, 2 with very strong support, 1 with more middling 0.76 probability support. But that's something we're going to be following up on, if maybe circoviruses themselves can tolerate some being packaged in the opposite orientation. This only works for Ambasense viruses because you have to be able to have a virion sense and a complementary sense in the same genome to do these calculations, but enough CREST DNA viruses are Ambasense that we think this is potentially a really useful complement to looking at that non-nucleotide region. So just to summarize what I've said today is that the CREST DNA viruses are a real group. They have the potential to have a true tree of life for at least one of their ORFs, the replication-associated protein. We have a matrix that we think resolves things best, and there are some differences between our trees created with our matrix and trees created with the general matrices. We don't know if the CREST matrix will ever be useful for anyone else's question, but it will be useful for our question, making the best rep tree possible. And we have two methods that are BLAST dependent, that don't rely on nucleotide sequence homology, that can help us classify viruses. Neither of them are supposed to replace what we're doing now, but we're looking for other complementary methods that can help us be more sure of our results, and more different kinds of independent sources of data that get us to the same place, make us more confident that we don't have to go through more taxonomic revisions. I'd like to thank especially our collaborators at the University of South Florida, Maya Breitbart and Karina Rosario, who are co-PIs with me on assembling the Tree of Life grant to look at the CREST DNA viruses. A huge number of members of my lab participated in different parts of the single-stranded DNA data sets that I've shown you here, but mostly Lola, Eric, and Alvin. I'm happy to take your questions, but if you like some of the things I'm saying, but wish it were all experimental work and in an RNA virus, maybe you know someone who wants to come be a wet lab postdoc with me on that career grant that was mentioned. Thank you.